Shri Gurubhyo Namaha children, I am happy to welcome you all for our online teaching academy, Ekalaiva. For the class 10, subject biology, unit 18, the name of the unit is Heredity. Today, we are going to discuss about types of chromosomes based on function. Yes, so based on the functions, what are the different types of chromosomes? The eukaryotic chromosomes are classified into two types. One is autosomes and another one is allosomes. Once again I am repeating, two different types of chromosomes. One is autosomes and another one is allosomes. Autosomes are nothing but, autosomes means body chromosomes. Yes, autosomes contain genes that determine the somatic characters, that is body characters. Male and female have equal number of autosomes. Okay, so equal number of autosomes. There are about 23 pairs of chromosomes, 23 pairs. Among these 23 pairs, 22 pairs are autosomes. Okay, so these 22 pairs are equal in male as well as in female. Okay, so the next one type one is allosomes. Allosomes are chromosomes which are responsible for determining the sex of an individual. The last 23rd pair only determine the sex of an individual. Okay. So allosomes are also called as sex chromosomes. So allosomes are also called as sex chromosomes. There are two different types of sex chromosomes are there in human beings. One is X chromosome, another one is Y chromosome. In male, the sex chromosomes are X and Y. In female, the sex chromosomes are XX. So the last 23rd pair, if it is XY means the individual is male. If the last 23rd pair XX means the individual is female. So the last pair only determine the sex of an individual. Clear children? The next we will see the karyotype. The number of chromosomes in an living organisms is constant. That means plants and animals. So the number of chromosomes are constant. In human beings, each cell having 23 pairs of chromosome. As I said earlier, each cell contains 23 pairs of chromosomes, out of which 22 pairs are autosomes, that is body chromosomes, and then the 23rd pair, the last pair, it is said to be the sex chromosomes or allosomes. In the body cells of sexually reproducing organism, the chromosomes generally occurs in pairs. As I said, chromosomes occurs in pairs. So it is said to be 23rd pair, 22nd pair. So totally 23 pairs. So chromosomes are usually occurs in pair. Okay. So this condition, the pair of chromosomes, this condition is said to be diploid. Diploid is nothing but 2n. Okay. The gametes produced from the individual, the gametes produced by the organism contains a single set, that is only one set of chromosome. So it is said to be haploid in nature. Haploid is nothing but n. Okay. So the gametes produced by an organism is having a single set, only one set of chromosome, then it is said to be haploid, that means N. So hence these gametes are said to be haploid, N. Karyotype, the word karyotype is nothing but, it is nothing but the number, size and shape of chromosomes in the cell nucleus of an organism. So karyotype is nothing but the number of chromosomes, the size of chromosomes as well as the shape of the chromosomes all together. So the number, size and shape of the chromosomes in the cell nucleus is called karyotype. Okay. The next one is ideogram, the diagrammatic representation of karyotype. Karyotype means what? The number, shape and size of the chromosome is nothing but karyotype. Okay. This diagrammatic representation of karyotype is nothing but ideogram. Ideogram is nothing but diagrammatic representation, only diagrammatic representation. Okay. It consists of all the metaphasic chromosomes arranged in homologous pair according to descending length. So they are arranged in descending order. The length, highest length place in the first place. Okay. So they are arranged in descending order, descending length, thickness, position of centromere, shape, etc. with the 6 chromosomes placed at the end. So 23 pairs of chromosome, as I said, the last pair only the 6 chromosomes, the 6 chromosomes are placed in the last place, it is placed at the end. So okay, the remaining chromosomes are occupied in the topmost place. They are arranged according to their descending length, thickness, position of centromere, shape of the chromosomes, etc. and then finally the sex of the individual, the last 6 chromosomes placed at the end. So the diagrammatic representation of ideogram, the diagrammatic representation of karyotype is said to be the ideogram. Okay. See the picture here, children. Each cell, a cell contains 23 pairs of chromosome, as I said earlier. The chromosomes are placed or arranged in a pair. See, this is the first one, first pair, and then second, third, fourth, fifth, like this. They are arranged in the descending length. Highest, topmost is it high length, increase in length, followed by very small chromosomes. Okay. So they are arranged in the decreasing in length, shape of the chromosome, size of the chromosomes. Okay. So see the picture here, all the chromosomes are occurs in a pairs. 1 to 22 pairs are body chromosomes. They are the body chromosomes. The last pair, so this is the sex chromosome. 23rd pair is the sex chromosome. If it is female means X female. If it is male means X Y male. Is it clear, children? So the last one only decide the sex of the individual. So what is ideogram? The diagrammatic representation of karyotype is said to be the ideogram. Okay. So what is karyotype? Karyotype is nothing but the number of the chromosomes based on the size, shape and the shape of the chromosomes. Okay. So karyotype is nothing but they are the arrangements, the arrangement of chromosome based on the shape, size. The next we will see the structure of DNA. It is very important question and very interesting too. Do you know very well, DNA is nothing but it is a hereditary material and it contains the genetic information. They are very responsible. It is responsible for the transmission of genetical information from one generation to other generation. Okay. It is mainly it is a most important constituent of all the chromosomes. So chromosomes contains DNA only. So it is the most important constituent of chromosomes. It is widely accepted model. The most widely accepted model of DNA is double helical structure and it was proposed by James Watson and Francis Crick. So James Watson and Francis Crick model is said to be the double helical structure, and this is the accepted model of DNA. See the picture here children, it is a double helical structure. See, one strand starts with 5' to 3' and another strand starts with 3' to 5' This is the double helical structure. This double helical structure is this structure was proposed by James Watson and Francis Crick, and it is the most acceptable model of DNA. Okay. Then chemical composition of DNA. What is made up of DNA? What are the components present inside the DNA? Let's see. DNA is a long molecule consisting of millions and millions of nucleotides. So it is made up of many nucleotides. So millions and millions of nucleotides. Hence it is also called as polynucleotide. Many nucleotide, as I said, no, millions of nucleotide. Hence it is said to be poly. Poly means what? Many nucleotide. So DNA is a large molecule consisting of, or it is made up of millions and millions of nucleotide. Hence it is also called as polynucleotide. See the picture here children. As I said, two helical strand. It is double stranded DNA, and most acceptable model of DNA is James Watson and Francis Crick model. So Watson and Crick model of DNA is said to be the double helical structure model. Okay. So here one strand running from 5' to 3' and another one running from 3' to 5'. Both they run anti-parallel to each other. They are the two helical structure. Okay. So this DNA is made up of millions and millions of nucleotide. This is the nucleotide. Hence it is also called as polynucleotide. Before going to see this chemical composition, first of all you have to know about what is nucleotide and what is nucleoside. Let's see. So each nucleotide consisting of three components. What is nucleotide? Nucleotide, it is made up of three components. One is sugar molecule, that is deoxyribose sugar. Second one is nitrogenous base, and third one is phosphate group. There are about 4 nitrogenous bases. It is divided into two groups. One is purine and another one is pyrimidine. Okay. So adenine, guanine or purine, and so these 4 are the nitrogenous bases. So what is nucleotide? Nucleotide is nothing but it is made up of sugar molecule, nitrogenous base and the phosphate group. So sugar means in DNA it is deoxyribose sugar. Nitrogenous base, there are two different group. It is divided into two group. One is purine, another one is pyrimidine. Adenine, guanine are purine, cytosine and thymine are pyrimidine. Okay. Then phosphate group. There are two different types of nitrogenous bases in DNA, two different group. As I said, purine and pyrimidine. Adenine, guanine are purine and cytosine and thymine are pyrimidine. See the structure of a nucleotide children. See, this is a nucleotide. It is having three components. One is sugar, second one is nitrogenous base, third one is phosphate group. Okay, sugar always it is deoxyribose sugar. Then nitrogenous base, two different groups, purine and pyrimidine. Adenine, guanine are purine and cytosine and thymine are pyrimidine. So this is the nitrogenous base and it is phosphate group. So all these are connected with each other to form a nucleotide. Nucleotide is nothing but sugar, base and phosphate. Okay. See, nucleotide and nucleoside. So what, first of all, you know about nucleoside. What is nucleoside? only the nitrogenous base with sugar, that is called nucleoside. Base plus sugar. How many bases are there? 4 bases. Adenine, guanine, cytosine and thymine. These are the 4 bases. So, adenine, guanine, cytosine and thymine. These are the 4 bases with sugar. What sugar? Deoxyribo sugar. Okay. This is called nucleoside. And then nucleotide means extra phosphate group. Just add one phosphate, then it is said to be nucleotide. Can you say the definition for nucleotide in another way? Please try. Shall I tell? Yes. The phosphorylated form of nucleoside is called nucleotide. Children, listen. The phosphorylated form. So, nucleoside is there, extra one phosphate will be had. The phosphorylated form of nucleoside is called nucleotide. That's all. The nucleotides are formed according to the purines and pyrimidines present in them. See the structure here, base and sugar, that gives nucleoside. In that, extra one phosphate will be added, then it is said to be nucleotide. Okay, the phosphorylated form of nucleoside is called nucleotide. Can you understand this? Shall we move? Yes. Now we will see the Watson and Crick model of DNA. DNA molecule consists of 2 polynucleotide chain. See the structure here, children. This is the one chain and this is the another chain. This is the one chain and this is the another chain. Both these are helical, double helical structure. It is a accepted model, that is Watson and Crick model of DNA. Okay. So, it is consisting of 2 polynucleotide chain. These chain form a double helical structure with 2 strands which run anti-parallel to one another. Anti-parallel to one another means one strand runs from 5'N to 3'N and another strand runs from 3'N to 5'N. So, it is anti-parallel to one another. The nitrogenous bases in the center, see the picture. This is the 2 helical structure, this is the one strand and this is another strand. In between these 2 helical structure, nitrogenous bases are present. These are the nitrogenous bases. Cytosine paired with guanine, guanine paired with cytosine, adenine paired with thymine and then guanine paired with cytosine. These are the nitrogenous bases. So, the nitrogenous bases in the center are linked to phosphate, sugar phosphate unit. So, sugar phosphate units are present at the helical structure, end of the helical structure. In between the helical structure, the nitrogenous bases are present. So, nitrogenous bases are at the center linked to the sugar phosphate unit which forms the backbone of the DNA. Okay. So, sugar phosphate units acts as a backbone for the DNA. So, in between this helical structure, nitrogenous bases are present. Pairing between the nitrogenous bases is very specific and it is always purine base pair with pyrimidine. So, adenine base pair with thymine with double hydrogen bond and guanine base pair with cytosine with triple hydrogen bond. Adenine and guanine are purine. Purine always base pair with pyrimidine, okay, with hydrogen bonds. So, this bonding is very specific. Purine always pair with pyrimidine. So, adenine base pair with thymine by 2 hydrogen bond and guanine base pair with cytosine with 3 hydrogen bond. Clear? See here, adenine links thymine A links with thymine with 2 hydrogen bond, and second one is cytosine links with guanine with 3 hydrogen bond. This bonding, this is called complementary base pair. Okay, so adenine, thymine, guanine, cytosine, this pairing is called complementary base pairing. Next one, hydrogen bonds present between the nitrogenous base make the DNA molecule stable. See, this is the hydrogen bond present in between the bases. This is the base, cytosine, and this is the base, adenine, base pair with thymine. Yes, so in between this base, what is this? Hydrogen bond. So this hydrogen bond between the nitrogenous bases makes the DNA molecule stable. Okay, each turn of the double helical structure is about 34 Armstrong. This is the one turn. So from here to here, this is a one turn. So the length of this one turn is 34 Armstrong. That is, in between this one turn, there are about 10 base pairs are present. So okay, there are 10 base pairs in a complete turn. One turn is nothing but this is the one turn. So the length of this one turn is 34 Armstrong. In this one turn, 10 base pairs are present. Clear? Then the nucleotide in a helix are joined together by phosphodiester bond. So this is the nucleotide, thymine, adenine, guanine, cytosine. These are the bases, and adenine, thymine bases with 2 hydrogen bond, and this is the nucleotide. These nucleotides are joined by phosphodiester bond. This is the phosphodiester bond. Okay. Thank you, children. To receive our online lessons, please press subscribe button and you will receive the latest updates. Thank you very much.